Hello everyone, my name is Maxime Malaussena, and today my presentation will be on how ethanol and acetate interacts with the gut microbiome. This is a review of an article by Cameron Martino and colleagues titled How Acetate Reprograms Gut Microbiota During Alcohol Consumption. Here's a short list of acronyms that will be used in the presentation. What is being asked? The article mostly tries to answer how does acetate levels alter any liver microbiomes when alcohol is consumed. Specifically, when ethanol is absorbed in the stomach or small intestine, travels through the bloodstream to the liver, what are the interactions between the small bacteria in the liver microbiome and the ethanol? And another side question that the author looks for is ethanol directly metabolized by the gut microbiome? Why should we care about this field? The liver is an important organ in the digestive system and has many small bacteria that the liver interacts with in order to fulfill its job. Understanding how ethanol levels impact acetate levels in the liver and blood can potentially allow us to fully understand how ALDs occur, whether from ethanol itself or gut microbiota that interact with the ethanol. And this field of research is important because the liver has many interactions with gut microbiota that need to be studied in order to develop new drugs that work with the microbes in the liver. How did the author go about answering this? The author used a multitude of experiments on mice, including changing their diet in test groups and including ethanol in order to test what changes it did to the liver tissue as well as blood tissue. Some other organic compounds were used, including glycerol and glycerol triacetate, also known as GTA. The gene sequencing methods used included shotgun metagenomics, metatranscriptomics, and metabolomics sequencing, which all are a form of next-generation sequencing in Illumina. Impact of ethanol and acetate on the body. Alcohol, when consumed, contains ethanol, which, once absorbed into the bloodstream, flows throughout the body and impacts multiple different organs, including the brain, the liver, and the stomach, which can cause changes in mood, irritations in the digestive system, increased heart rate, and sweating. Acetate, which is produced in the liver when the liver absorbs ethanol, is used in the production of acetyl coenzyme A, which is important for energy production. Alcohol consumption. Alcohol abuse is a big social problem that many people face, or many people who know people face it. In 2023, 15.1% of young US adults met the criteria for past year alcohol use disorder. In 2022, US adults spent on average $583 per person on alcohol. And in 2021, the US adult per capita consumption of ethanol from any alcoholic beverages was 2.51 gallons, which is roughly 535.5 standard drinks per year. Now, this is definitely impacting older generations because Gen Z is spending much less on alcohol than previous generations, but this still impacts everyone. So what is the need and urgency of this problem? Well, alcohol can lead to many liver-related diseases. Alcohol use disorder in the US can lead to alcohol-related liver diseases, which impacted 28.1 million adults aged 18 or older, including 16.6 million men and 11.5 million women in 2023, which is 10.9% of the US population over 18 years or older that year. In 2023, 4.5 million US adults were diagnosed with some form of liver disease, with many millions of more adults having some form of disease that hasn't been diagnosed yet. And most liver diseases can lead to cirrhosis, which is the final stage and usually uncurable. Cirrhosis is the 9th leading cause of death in U.S. adults, resulting in 52,222 deaths in 2023. Understanding how the liver microbiome works with the liver could help scientists prevent ALDs from occurring. What did the author want to answer? The authors have a few questions that they are trying to answer with this research. They want to specifically know first, how does ethanol impact acetate levels in the gut microbiome? They also want to know, does the gut microbiome metabolize ethanol on its own, which they hypothesize that ethanol is co-metabolized due to the presence of certain organic compounds in sisal content. And the last question is, are gut microbes a direct cause of alcohol-related liver diseases, which their hypothesis is changes in gut microbiota related to ethanol consumption may be a cause for alcohol-related liver diseases instead of ethanol itself. Methodology. The authors want to first test to see if ethanol has any impact on acetate levels. The authors do this by giving mice a diet consisting of ethanol plus nutrients over the course of a few weeks and then collecting their tissue samples at the end. The authors first administer a little bit of ethanol day by day, starting with 10% on days 1 to 3, 20% on days 4 to 6, and so on. Eventually, on day 15, oral galvage with ethanol is used twice a day on day 16 and 17 and the morning of day 18. And on day 18, liver, plasma, and cecum tissue is collected via Blythe and Dyer-based extraction. For the author's second experiment, they wanted to test if ethanol is metabolized by liver microbes. Mice were fed a liber dicarylate diet for a week to get acclimated. Then they were split into two groups, one control group which continued the same diet, and the test group which had ethanol included into their diet. After 8 more weeks, the mice had their tissues collected, their plasma and cecum tissue, using a modified Blythe and Dyer-based extraction, which were then sequenced. The third experiment of the authors use is a continuation of the second experiment to answer whether ethanol is metabolized by liver microbes. Similarly to the second experiment, mice will acclimate on a specific diet, but this one consisting of glucose. On day 8, mice are divided into two groups, one group adding glycerol to their diet and the other group adding GTA to their diet. The group with GTA were given increasing dosages over the days. At the end of the 2-week period, liver, cecum, and blood samples were collected from these two groups. This experiment is then repeated, but with an ethanol-based diet instead of glucose, with 29% ethanol. And once the groups are divided into a glycerol and a GTA group, the group with the GTA administered did not have dosages increased. And once again, at the end of the 2-week period, liver and cecum, blood samples were once again collected for sequencing. The sequencing methods used by the authors included shotgun metagenomic sequencing using a Nextera XT library sequenced by an MI-seq 2x300 BP cycle paired ENKit, which is next generation sequencing done via Illumina. And the other sequencing they use is metatranscriptomic sequencing using the same library sequenced by an MI-seq 35 BP cycle single ENKit, which is also next generation sequencing done via Illumina, which we discussed in class. Results. Does ethanol increase acetate and acetyl coenzyme content in the liver? So from the first experiment, the authors got data on a certain carbon carbon molecule called palamitate, which is a 16 carbon chained molecule, and it had one of its isotopes thoroughly outnumbering the rest of its isotopes, meaning that palamitate was being metabolized way more often, 8 times more often than mice with not injected with ethanol, meaning that more acetate was being synthesized from ethanol because acetate does synthesize into acetyl-CoA enzyme. The authors also found out that in the content of the plasma, liver, and zircon, there was way more acetyl-CoA enzyme percentages, with 28% in plasma, 24% in liver, and 18% in the zircon content on average. They also looked in the TCA cycle in the liver with specifically the intermediates that are formed in the cycle, and they found that slightly more intermediates were being formed with 5% of citrate, 6% of alpha ketoglutarate, 3% of succinate, 4% of fumarate, and 3% of malamate, which is 2% or 3% more than what it is normally in the liver. And while not mentioned here, is more of the acetyl-CoA carboxylase inhibitor was found in the content to decrease the amount of palamate synthesis being created approximately by minus 0.5%. So the authors did concluded from this first experiment that ethanol is in fact contributing to the increased amount of acetate and acetyl-CoA enzyme in the content in the ethanol ingested mice. Is ethanol directly metabolized by gut microbes? So for the author's second experiment, they wanted to see if certain liver microbes, specifically the bacteroidetes, are impacted when alcohol is ingested into the liver. So in the figure 2b, you can see for the ethanol group of mice that there is an increase on average of the number of bacteroidetes relative to enterococascase microbes, which is another microbe found but not impacted by ethanol in the liver microbiome. And they found that there was increased numbers of bacteroidetes when ethanol was added. Now, there were no changes to the amount of ethanol converted in ethanol-fed mice from ethanol to acetate, because the process going from ethanol to acetate is done by the liver host cells instead of external bacteria in the microbiome. But, however, they did see that when more ethanol was added into the system in figure 2D, that gut microbes were responding to the increased ethanol by increasing acetate dissimilation, which in turn would decrease the acetate levels, which would cause the liver host cells to increase the amount of ethanol they metabolize into acetate. And they did check for all of the fatty acids, the small chain fatty acids that were in the liver microbiome, but only acetate was the one that had a difference in concentration, specifically being 4 times increased in the ethanol-fed mice versus the control group, compared to all the other groups of the SCFAs, which did not have any significant changes. One specific detail that the authors did find in the second experiment was that gluconeogenesis was found in higher rates in ethanol-fed mice compared to the control group. And Gluconeogenesis is a process in which glucose is created from small fatty acids such as acetate, implying that bacteroidetes were upregulated due to the increased amount of acetate. So the authors then concluded that bacteroidetes may play a role in the metabolism of acetate instead of ethanol, explaining why they are found in increased amounts in alcohol consumption due to the increased amount of acetate instead of ethanol. Continuing from the second experiment, the authors wanted to see whether acetate levels on their own impacted the gut microbiome. They did this by using glycerol triacetate, as otherwise known as GTA, to increase the acetate levels in the microbiome without having acetate being metabolized from ethanol. So they gathered 4 groups of mice with glucose and ethanol, as well as then dividing them again into glycerol and GTA to impact their ethanol and acetate levels. And in figure A, you can see that only the acetate levels were impacted instead of any of the other short-chain fatty acids. And this is the acetate levels being synthesized by the ethanol. They also noticed in figure D that no liver damage was spotted in the glucose groups, meaning that liver damage is primarily caused by the metabolism of ethanol to acetate instead of just acetate itself. However, they did find a similar abundance of the microbiome in the glucose GTA group that was similar to both the ethanol feds groups, meaning that the amount of acetate did impact the number of microbes in that liver biome. And they also found that the bacteroidetes enrichments relative to the enterococave were similar in both the glucose GTA group as well as the ethanol glycerol group, meaning that ethanol did not have an impact on the microbes, but instead it was the acetate. Therefore, they concluded that the metabolism of ethanol was not significantly impacted by the GTA group, and instead it was just the level of acetate. Discussion, results interpretation. So the authors did conclude that there is an impact in acetate levels due to ethanol because of the metabolism of ethanol by the liver host cells to acetate. They did also see that increasing the acetate levels induces a similar change as intaking ethanol on the gut microbes, but the gut microbes that metabolize acetate were not sufficient enough to be considered the cause for any liver diseases in the absence of ethanol and just the GTA group. They also concluded that ethanol is not directly metabolized by the gut microbiome, but instead metabolizes the acetate into the acetyl-CoA enzyme that is created from the liver, metabolizing ethanol to acetate. And they also concluded that acetate might play a key role in microbiome alterations induced by alcohol consumption. But further studies with alcohol as well as a GTA modified microbiomes would be needed to see whether the acetate levels impact other microbes besides bacteroidetes. Conclusion. The authors concluded that ethanol is not directly metabolized by gut microbiota, but instead ethanol levels are impacted by the gut microbiota metabolizing acetate, causing liver cells to metabolize more ethanol. They also concluded that detrending the effects of acetate may help us understand what changes in the gut microbiome occur and what changes from that can cause alcohol-related liver diseases. A couple questions I want to ask to the group include, what further studies could be done to understand the full impacts of acetate on gut microbes? I also want to ask, should other methods of gene sequencing besides NGS be used to understand acetate's impact on gut microbes? You could also consider, how would you change this experiment to test for any relationships between ethanol and the gut microbes instead of the acetating gut microbes? As well as, outside of the experiment, what is your ethical stance on testing mice? And a final question is, should research funds be used for research for alcohol-related diseases when we already know alcohol is bad for your body, specifically the liver? Meaning, should we spend money on this research when we can just be using a PSA for using alcohol, so we can save money for research such as cancer research or other very important research that affect even more people? Here is my list of references, and thank you so much for listening to my presentation.